starting to think about, probably just in 2019, was if there's an outbreak of a new virus, how are we going to produce a new vaccine really quickly? So with this delivery system, you can adapt to make many different vaccines. We'd only ever done it in a very slow fashion. We'd only ever made a batch and tested it in the lab, and then applied for some funding, and then manufactured it, and then maybe 2 years later done a clinical trial, going very, very slowly. And we knew that if there was an outbreak of a new virus, that was much too slow. So we started to think about what would we do if there was a new virus that we've never seen before that starts to infect people. We need to make a vaccine. What are the individual technical steps we need to go through? Get a vaccine ready for a clinical trial as quick as possible. So instead of taking 3 years from thinking about it to actually be able to start the clinical trial, how quickly can we do that? And I applied for some funding to develop this further and wasn't successful. But at least
something you couldn't even raise a few $100,000, I think, right?
No. So, but at least in writing the grant application, I'd gone through the thought process of, well, what would we do? And so then what happened, it was actually on the 1st of January 2020 when I started to see some news of cases, 4 cases of pneumonia, unknown cause, suggestion that this could be a new virus. I was interested in it because I've been doing this work on thinking about if there is a new virus, how can we make a vaccine really quickly? So it was the right timing for me because I'd already gone through that whole process and then see these reports and this could be a new virus. Probably is, and it's probably influenza or it's another one that we know about. It could be SARS. People were worried about SARS because there's been an outbreak of the SARS virus, which is a coronavirus, some years ago, and this had quite a high fatality rate, and people have always been concerned that it may come back. So was this SARS making a return visit? And so I was watching the story unfold over the first few days of January 2020. There were 4 cases, then there were more cases, then people were in hospital, people were dying, and it wasn't influenza, and it wasn't an adenovirus, and it wasn't SARS. We still didn't know what it was, but it was getting more and more interesting to me as somebody who had been thinking about a new virus emerging, because it was getting more and more likely that it was a new virus, and eventually we did get the information then that it was a coronavirus. And it's a new one, it's not one we've seen before, it's not SARS virus, but it's closely related. So fortunately for me, it was a coronavirus because I've been working on the MERS coronavirus vaccine. So there are lots of different families of viruses, but it's helpful when we're thinking about our approach to dealing with making a vaccine or therapeutics to know which family that virus is in, because it will have similar characteristics, and we can deal with them in a similar way. And because I'd already started to test a vaccine against MERS coronavirus, and we'd done 2 clinical trials by that point, we knew that the technology looked like it was going to work really well for this. We were getting very good antibody responses, the vaccine was safe. We'd only tested giving 1 dose to people at that time, but it looked like it was the right approach to take. And the technical details of exactly how we designed the sequence that we put into our vaccine, we've been through that. We worked through it all for the MERS vaccine, and so when it started to look like this is spreading and we should be doing vaccine development, I knew that if we were going to test a vaccine in a clinical trial, I couldn't make 10 different versions and see which would work best. I was going to have to pick one because we have a very small manufacturing facility on our campus in Oxford where we can make vaccines and then get them approved to go into clinical trials, but we can only do 1 at a time. And we would only have the capability to test 1 vaccine. So for me, it was a question of what is the one thing that we will test? We'll copy what we did with the MERS coronavirus vaccine, because that actually looked like it's working really well, so don't change what's working. Repeat the same approach for this new vaccine. But even then, when I started working with colleagues, and lots of other people were interested at this time as well, started making the vaccines, tested in the research lab, it was still with the idea that this may never be needed. This, it's a small outbreak. It may be wrapped up very quickly. There's a lot of surveillance. People know where the infections are. We may never need a vaccine. Actually, obviously, that was not true. We carried on, knowing that if we did need a vaccine, we need it as quickly as possible. And it's good that we did continued to push forward as fast as we could, because what then became apparent was that the virus had spread much more than anybody recognised. It was across Europe, a lot of cases in Italy. But I remember the first few cases being detected in the UK. There was a lot of press about, the first 2 people in the UK with the COVID-19 with SARS-2 virus infection. I think we didn't even have that name at that time. And then there was a family who'd been skiing in Europe, and they came back, and some of those were infected. And then all of a sudden, just everyone. And what we realised was that we hadn't been detecting it, the diagnostics weren't there, the surveillance wasn't there. And so, yes, this was spreading, and this is now much bigger than we thought it was going to be. So an outbreak is something that happens really just one location. It may spread through a few countries, as it did in the Ebola outbreak of 2014, but it's all linked within a defined geographical region. But a pandemic is when you get multiple countries involved, and then it's around the world. And then, it's out of control. We have no possibility of stopping the spread. We have to work out how we deal with the spread of virus, rather than thinking about can we possibly stop it. And we were still, people were still taking measures to try to slow down the transmission of the virus, and try to slow down the spread, because that gives us more time to work out what we're going to do about it. Can we get vaccines in development? Can we find out what existing drugs work? So we didn't want to just let rip, but on the other hand, it was known that this was going to go around the world, and we couldn't stop that, so we had to get our response ready.
And you're thinking on, first of all, how to kind of then accelerate it. So you've developed the kind of science of the vaccine, you've done the small batches. But what was your thinking in, okay, 4 locations, 24,000 people, how do we speed the clinical trials up? And literally the idea of not taking any shortcuts, just have to speed it up. That's my one question. The other one was to do it at scale, how to financially do it, whether to get companies involved or consortiums involved. Maybe you could kind of explain a bit about that journey.
Okay, so what happened was that we started accelerating the vaccine development. We stopped some work that was happening in our own vaccine manufacturing facility. It was a vaccine being made for one of my colleagues. I said, just leave that, that's not important, that's not the priority now, we'll stop. We'll start manufacturing this vaccine instead, and we're able to do that because have control of what's manufactured in that facility. We've got people involved in planning the clinical trials. So Professor Sir Andrew Pollard, as he now is, I talked to him about getting the clinical trials underway. And I was thinking about the first clinical trial, but he took it much further than that, because one thing that we learned in 2014 is that we had to go much further than just a little bit. We didn't want to be just doing the first part at the beginning and then expect to hand on to somebody else, because that hadn't worked. We had to take this as far as we could. So he was able, with a network of clinical trials.
So you really did feel that you had to have control as far as possible?
Yeah.
Because it was very difficult to kind of hand it over.
Yeah. We had to take this as far as we could. And so from my thinking about, let's start the first clinical trial in Oxford, he then had a network of 18 clinical trial sites around the UK, and then started going international with the clinical trials. So we had some in South Africa, big clinical trials in Brazil, so Kenya as well. So we knew we could, as a team, get this vaccine through into the phase 3 trials, which is where we see if the vaccine actually works to protect people against the infection, stop them having the disease. But we're a university, and so we're never going to be able to manufacture the vaccine at scale. We could organise the manufacturing of the trials, but we weren't going to be able to organise the manufacturing of the vaccine for the use once it gets licensed. We need a company to work with to do that. We need a pharma partner, and we need somebody who's going to think big.
And for the capacity and the money.
Capacity and the money, and also the willingness to work with us to do the vaccine development the way that we had always wanted to do it. So as a university, we're a charitable institution. We're not out to make money from this vaccine, and we want it to be used as widely as possible. So we always said we want a vaccine for the world. This is not a vaccine for the UK or a vaccine for Europe. This is a vaccine for the world. Going back to developing this technology, we worked on this technology because it was suitable for very large-scale manufacturing and use in many different geographical locations. It doesn't need the very low temperature storage. That makes it much easier to feed into existing vaccine distribution networks. So that's a good feature of the technology, apart from the
And it could be manufactured at low cost to keep the cost.
Yes. So AstraZeneca became our partners and they agreed to produce the vaccine without profit during the pandemic and without profit in perpetuity for low and middle income countries. So once the pandemic ended, they were allowed to make some profits in high income countries, but not in low income countries. And so because they weren't making a profit and because it was suitable for very large scale manufacture, it meant that the price was kept very low. And that really helps with making the vaccine much more accessible, because somebody has to pay to produce the vaccines, and the cheaper the vaccine, obviously the more vaccine you produce for the same amount of money. So that was really important. But the other thing that AstraZeneca did that was enormously important was start thinking from the very beginning of our partnership in the late spring of 2020 about how they would make large quantities of this vaccine. And they had to invest funds.
That was actually not that much after, because March is when kind of the whole world sort of woke up and started shutting down.
Yeah.
So you're literally talking about, very fast work.
Yes. Yeah. So what they could have done under the terms of our agreement with them is said, yes, we'll take this on, we'll manufacture this vaccine, we won't make a profit, but then just worked with one or two different manufacturing sites and made a small quantity of the vaccine. But they decided not to do that. They went way beyond that, and they worked with many different manufacturing sites around the world. So they invested a lot of money and a lot of time to get the manufacturing underway while we were still doing the clinical trials. So this was this is what we talk about by working at risk. Their money is at risk because we're testing the vaccine to see if it's effective, and they're already planning to manufacture it in billions of doses. And if the clinical trials had said no, it doesn't work, all that money's wasted.
So as a scientist, I mean, I remember, I think, hearing an interview yours in the early days, and you were pretty confident about. So was that based on because of your MERS work, or what kind of things gave you that confidence?
It was based on all the work that we'd done before. So the MERS vaccine trial wasn't the first vaccine trial we did with that technology, it was about the 12th. We've used it in many different situations, but particularly having done the MERS clinical trials and knowing that with the coronavirus, we get a strong immune response with a single dose. And having tested this technology in people of different ages previously, so in the flu vaccine trials that I've done, we tested it in people up to the age of 80. And we got very good responses. And in malaria vaccine trials, it's been tested in infants in Africa, and they got very good responses.
So you were confident on the delivery system?
Mm-hmm. Confident on the delivery system, but then specifically because of the MERS vaccine work that we've done, knowing that the way we've made the vaccine against SARS-CoV-2, against the spike, that this was highly likely to work. And I felt, well, if this doesn't work, I don't know what will. There's always a possibility that it won't be very effective, but I didn't really see what else anybody could do that would be a great deal better if this failed. But what happened in the UK is that the government set up the vaccines task force, which was led by Kate Bingham, who's a venture capitalist, and she's used to assessing different medical technologies at an early stage of development and deciding if she wants to invest. So she's used to looking at, is this likely to work? Is the team good? Is everything in place? And so she was tasked with assessing all of the different vaccines that were in development for SARS-CoV-2, for COVID-19, in spring of 2020, and making assessments about which to invest in. And she, very wisely, decided to invest in many different technologies. So somebody told her that only 1 in 7 vaccines was likely to work. So she said, well, we have to invest in at least 7 then to have any chance of them working. But she set up a team that could assess the information from each of the vaccine developers. What have they done? What are they doing now? What are the prospects for wide-scale use? And deciding where to invest. And so at that point, we actually then did start to get investment from the UK government, because ours was one of the technologies that was chosen to be supported by the UK Vaccines Task Force. And in fact, they did a great job, because everything that they decided to invest in was successful eventually. Some of them took longer than others, but they picked winners, they picked a portfolio of winners. From our point of view, we then had the investment so that we could get our clinical trials running. We were working with AstraZeneca to explain to them how to manufacture the vaccine. They took that on, and then they transferred that knowledge out to many different vaccine manufacturing companies around the world. But each time they did that, they would spend months working with each of those manufacturing sites, which already had to have the facilities. There was no time to build new facilities. They had to have the facilities, they had to have the trained staff, they had to have the experience of manufacturing vaccines to extremely high quality and being able to test them to know that they were very high quality. And then there would still be months of communication needed to get the manufacturing up and running at each of these sites. But it was because they did that we were then able to see 3 billion doses produced by the beginning of 2022. Serum Institute of India did a great job. They produced probably about half those doses. They always work on a very big scale. Some of the other sites produce less. But for example, in Brazil, there was a company there that hadn't manufactured a vaccine anything like this one before. So initially, they just took on the task of receiving large quantities of vaccine that had already been manufactured, but their job was to put it into the vials so that it could then be distributed. And they did that, and that's quite complicated. But then they wanted to do the whole process. So then they received the technology for the whole manufacturing process. And so they were then able to manufacture vaccines in Brazil, to use in Brazil and in Latin America more generally. And we really like this approach of manufacturing for local use, because that's how you get vaccines around the world much more quickly. When you need to work really very fast, that's a very good way to do it.
So take us. So basically, the AstraZeneca is investing a lot in producing all these producing a huge quantity of vaccines, but at the same time, your results are coming in for the clinical trials. So I think you've got the so from early spring 2020, and then you started to get the results in, and just tell us a bit about that time when you're getting the results from the clinical trial, because that's like a key moment of the future of mankind kind of thing.
Well, it was really frustrating for us waiting, because the trials are placebo-controlled, randomized, double-blind. People don't know which vaccine they've had. Did they have the COVID vaccine, or did they have the other vaccine that we were giving them, which wouldn't have protected them against COVID? And then we have to wait until enough people, through just doing what they would normally do, who've been infected, and each person in the trial who is infected, their results have to be looked at very closely to make sure they fulfill all of the precise criteria for being what we call an endpoint in the clinical trial. Yes, this really was an infection. It was their first infection. They didn't have any antibodies to COVID when we first immunised them, and we couldn't check that at the time that we started, so we had to do it retrospectively in many cases, because we were working so quickly. And we didn't even know how many cases there'd been, because only the statisticians know that. So everybody else just has to sit and wait, and what we're waiting for is to get to a defined number of cases that have happened in the trial, because the statisticians know that when we get to that number, they will analyse the results and see how many were in the group that had the COVID vaccine, how many in the group that had the other vaccine, and that will tell us if the vaccines work. So we're in the dark. We're just waiting to find, are we there yet? Are we there yet? Why is it taking so long? In the UK, we recruited people over the age of 18. We started with people 18 to 50, and then we expanded into older populations. The ethics committee wanted us to go quite slowly, so not immediately immunise large numbers of people in trials who were over the age of 50, but we were able to add them in. We didn't have many infections in that age group, because those people were obeying government advice and staying at home and protecting themselves, which is what everybody should be doing. But that meant that it was very difficult to know if vaccines worked in that age group during the clinical trials, because there weren't very many infections in that age group. So when we did finally get the data, and it was unblinded, so you may not have noticed, but what happened was every time an announcement about vaccine efficacy from a COVID vaccine trial was made, it was always on a Monday morning. And that wasn't coincidence. It's because companies are obliged to report results as soon as they know them before the market's open, so that there can't be any kind of dodgy dealing on selling shares and so on. So everybody would wait until Friday before they then start to look at the data and see what it is. So you've at least got the weekend to analyse it, but then you have to announce it before the market's open on Monday morning. So there was a weekend where we were told it's going to come this weekend, and we'll know what it is, and we were waiting to see. And we actually got the news that it was a bit more complicated than we'd expected. So whereas other vaccine trials had announced just 1% for efficacy, we actually had different percentages in different groups of the trial, and it took us a while to understand why that was. But it was actually really interesting and really important, because what we found out when we had a chance to look into the data further was that people who'd had a longer interval between their first and their second dose of the vaccine had better protection. And that's because they had a stronger immune response. But this was really important to know in the vaccine rollout, because it meant that the right thing to do was to give people their first dose of the vaccine and then wait for 12 weeks before they got the second dose. They would have a better result themselves, but it also meant that more people could receive their first dose. So in some countries, using other vaccines, the vaccination centres were holding back doses because they wanted to be able to give people a second dose at 4 weeks. And so they wouldn't give the first dose to other people. Whereas in the UK, the decision was taken to give people the first dose of the vaccine and come back after 3 months to get the second dose, so more people could get the first dose. And then what happened, because there was very good data collection, was that by March, very early March of 2021, data started to come through on the efficacy of the vaccine in the first people who started to receive it. So that was the people over 80. So this was an age group that we didn't have a lot of information on in the clinical trials. We knew the vaccine was safe, we knew we were getting immune responses. We didn't actually have data on how effective it was. But as soon as the vaccine rollout started, we very quickly got the information that it was highly effective, even in people over the age of 80. It was stopping them from going to hospital, it was stopping them from dying after their first dose, not even after the full vaccination schedule. And then they started.
That's like the most kind of riskiest group.
Yes.
Yeah. So, just to kind of take stock of, like looking at vaccinations and more developed countries and developing countries. The problem of like a huge amount of the developed world got vaccinations, but large parts of the developing world didn't. Perhaps you could talk a bit to the kind of challenges there.
Well, I mean, so despite the fact that AstraZeneca had set up manufacturing in many, many different sites, it still takes a while to ramp. I mean, this is a massive effort to go from never having made this type of vaccine before to making huge quantities of it in just the space of a few months, but also having to do it to very strict quality standards, meant that it did take a while to get large numbers of doses coming through, which did eventually start to happen. And then governments would be controlling where those doses were going. So the UK wanted their vaccine because they'd invested in it. In India, at some point, the government put an export ban on the vaccine because they decided they needed the vaccine for India, and then it wasn't until that was lifted that more could be supplied into the WHO-led COVAX program, which was arranging distribution of vaccine for other countries. And so it was frustrating to see that we couldn't get the vaccine to everybody quickly enough, but I think if this was to happen again, we'd be better prepared next time, because all of these systems are now in place for manufacturing vaccine in lots of different parts of the world. But this was the first time this was ever being done, so it took a while to get going. Then what happened was we came to a point where there was enough vaccine, but some countries were finding it difficult to use the vaccine as fast as they could be provided with the vaccine, because in many countries, although there's a good childhood immunisation programme, there isn't a routine programme for immunisation of adults. So that had to be created, and that is difficult to set up and expensive to set up from scratch. It took a lot of the healthcare budget to start to get the vaccine used across the population. And in some countries, healthcare workers were well vaccinated, largely because they're more accessible. If you work at a hospital, it's easy to get the staff in the hospital to come in for vaccination. But if you have a population living in a rural area, maybe some distance from that hospital, it's much harder to reach them. And so we were seeing that although vaccine could be provided, not every country could use it at the rate it was being provided.
And so, you were sort of saying previously that you couldn't get that much funding, even though there was always the fear that a pandemic would happen. And what are the learnings from this? And are you getting more funding now?
Well, we're getting some funding. We've still been struggling for funding, ironically. I've been trying to return to the programs I was working on before the pandemic because those programs were stopped. So Nipah virus, which is really important for Bangladesh, one of the reasons I wanted to come here. Lassa fever virus causes cases in West Africa, MERS coronavirus in the Middle East. And MERS particularly could cause a pandemic because it's another coronavirus.
So you do think that there is a serious a risk of another pandemic happening?
Absolutely, and there's always, sorry to worry people, but there's always going to be another pandemic. And what we need to do is to make sure that we're as well prepared as possible, so that when it happens, because it inevitably will happen, we can respond much better next time, and we can have a better outcome. If we can stop an outbreak from becoming a pandemic, that will save so much money for the economy of the world that it's really worth investing in trying to do that. So there's two main approaches that we want to take. One is the viruses, the pathogens that are out there that we already know about. We should be making vaccine stockpiles, we should be testing them, we should be ready, so if there's an outbreak of MERS in the Middle East, we've got the vaccine ready to use and contain the outbreak. The other thing that we should be thinking about is new viruses that we haven't seen before, and getting our systems in place, surveillance, but also how will we make a vaccine more quickly next time if it's a new virus. And what we can do to help with that is, if we have already spent time developing vaccine against each of the different types of viruses, one example for each of the virus families, so that we know what works for each of the virus families. Then if a new virus emerges, we need to know, well, which family does it fall into? What's been done already for viruses in that family? So this is the approach that is successful. This is what we will do for the new one. We didn't know in 2020 if these vaccines were going to work, because we don't have vaccines against any other human coronaviruses. So we were making our best efforts and predicting that this was going to be successful, but it wasn't until we started to see results coming through from the phase 3 trials that we knew for certain that yes, this really does work.
But you did have information on other coronaviruses which had to say.
Well, we only had information about the ability to induce an immune response. We didn't know if that immune response was strong enough to protect people. So it could have been that we get a low antibody response and you need a really high antibody response and it wasn't enough. Fortunately, that wasn't the case. But we haven't completed the process of testing the first vaccine. We'd only done the early stages. So we had to wait for the phase 3 trials. And yes, you can make a successful vaccine against a coronavirus infection. There are a couple of vaccines against coronaviruses that infect animals. One that infects birds, and one that infects poultry, chickens, and one that infects calves, and they work. So there was some precedent.
So was that spike protein?
Those were live viral vaccines, not the type that we want to use in people.
So let's say looking forward, the world having gone through all of this, and you're sort of saying there should be research and money invested into doing stockpiles of vaccines against infectious diseases that we already know about in each of the families, and then have research on potentially families that potentially might give rise to new pandemics. So 2 questions around that. Is there investment coming in now? And the second thing is, countries like Bangladesh, who, we're taking rudimentary steps in research, but there's a very enthusiastic and engaged, young group of researchers who would like to see Bangladesh being involved in research of vaccine development and vaccine production. What is your, first of all, about the funding, and then what is your advice?
Okay.
For development, I think you're doing some work in Brazil, so maybe that.
Yeah. So In Oxford, it wasn't just the vaccine development that we did. We also had the recovery trials led by Professor Sir Peter Horby and Professor Sir Martin Landry. Peter's arriving in Bangladesh today, I hope, if travel goes well. And those trials tested existing drugs on patients hospitalised in the UK with COVID to see which existing drugs worked. And they found out very quickly that dexamethasone is effective, it's cheap, it's widely available, made a massive difference. And we had other people in Oxford doing lots of other things, serology testing, measuring antibodies across the population. So there was a lot of effort went into pandemic response in Oxford. So we decided to keep these group of people working together. We now have the Pandemic Sciences Institute, which is a new institute in Oxford led by Peter Warby, and I lead the Emerging Pathogen Vaccines group within the institute. And so we are trying to raise funding for building the Pandemic Sciences Institute, but also to support our research in all of these different programs. So it's not just vaccines, it's all aspects of pandemic preparedness and pandemic response, including the social sciences. We don't have time to go into it, but a lot of interesting work on why people sometimes don't want to be vaccinated and how they behave when they're asked to go into lockdown, all of that kind of thing. So we are trying to raise funding for that. And I can't honestly say that we have all the funding that we need. We have some funding has come in, but we still have to continue to raise funding to support that work so that we can do more of this in the future. But one thing that we learned is that collaboration and networks are really important. So the Pandemic Sciences Institute is part of a network of institutions in the UK that will also work on aspects of pandemic response. I think we probably have the broadest range of research in Oxford, but we work with other groups in the UK who work on particular aspects of surveillance or diagnostics and so on. And that's something that was really important in developing the COVID vaccine, the collaboration between different groups, the collaboration between academia and industry, between the UK, Brazil, UK, South Africa, and bringing together people of many different disciplines. So my team did the very early vaccine development and the early vaccine manufacturing, the people in the clinical trials, the first clinical trials, and then moving on to very large clinical trials and working out how you immunise large numbers of people very quickly. So I think what's important for a country like Bangladesh is to think about how to set up these networks and how to become involved in existing networks. So to be part of this collaborative approach. We know that academics often like to work fairly in an isolated way, and that they don't, they're worried about competition. But actually what works much better is if we collaborate with many other groups and we team up and we work out what is our particular piece of the puzzle that we need to complete and how can we contribute to this whole effort. And so collaboration between companies that make vaccines and academic groups that can work on the design of new vaccines or the testing of new vaccines, that the CROs that can run clinical trials, how can all of these work together to put together the whole puzzle so that we can actually get more vaccines into development. There's no money in this. Nobody's going to make money out of outbreak pathogen vaccines, because a successful vaccine against a vaccine that causes outbreaks will actually be not used in very large quantities. It's a success if we manage to vaccinate a few people, contain the outbreak, and then we don't need any more vaccine. So it's not a money-making initiative. So we have to think about how can we get the funding to do this for public health benefit. But that means we have to be really efficient in the way that we use that funding. And so it's not going to work if people are individually trying to do the same thing in isolation, in competition. We need to put together the teams and the networks that can work together efficiently and quickly to get all of this work done.
Well, I think it's time for some questions now. Can we have the lights on, please? Yeah. Right. So, the maroon jacket.
Good afternoon, everyone. My question is actually two questions. First of all, if you see the drug development industry, it takes almost 12 to 15 years to develop a successful drug with $1 million cost, and still 90% of products or projects got failures. So what hurdles do you think can we take so that the time and money become less and the success rate goes up? high to develop a vaccine or drug, some products like that.
So how can we develop vaccines or drugs more quickly?
Yes, ma'am.
And also
Reduce the cost
the lower cost.
Success rates
So-
also increase.
Yeah, so, I mean, the way we were able to go quickly in the pandemic was because it was everybody's top priority. So the regulators did their job very fast because instead of us providing them with a very large dossier of information for them to assess once we completed the dossier, they took each piece of information as it was completed. So by the time we got the last piece of information that we needed to supply to them to get permission to start clinical trials, they'd already reviewed everything else. But they can't work like that all the time. So having a strong regulatory authority that can work in an efficient way is really important. Having ethical committees that are competent to review clinical trial proposals and again do that efficiently is really important. In some places, it takes a year, maybe 18 months to get approval to start a clinical trial because there are just processes that have to be gone through. And they don't need to be that slow. I mean, we can't do everything as quickly as we did in 2020, but we can speed up some of that. And if you're having to wait a very long time to start your clinical trial, that just ends up adding to the cost. So I think, working with the regulatory authority and the ethics committees to think about how can we do the complete process of approval, but do it in an efficient manner, is something that's going to help with future vaccine and drug development.
Yes, front, yes, right.
I'm Kaiser, Renata Limited. I had a two-part question about the dosing regimen. One was, did you have any a priori research that indicated that the 12-week gap between the two dosings would be optimal? That was one. And the second, I vaguely remember that in the dosing regimen, the half and the full was slightly fortuitous discovery. Rather, even though the science of giving a low dose so you don't have a massive antibody response was sound, but apparently this half bit was done just came out, it was luck.
Yes. So most of the work we've done on before 2020 just looked at using a single dose of the vaccine. We haven't been looking at 2 doses, and so we haven't got into the whole cycle of thinking about what should the interval between the doses be doing, because we've just been thinking about, for an outbreak, you need a really strong response from the first dose. We don't want to develop something where there's a very long vaccination regimen. That was our original thinking about the COVID vaccine trials, but I also wanted to know what would happen if we gave a second dose, because as I was saying, we don't know how strong the antibody response would need to be protective. We knew we could get an antibody response, we didn't know it would be enough after 1 dose. So in the very first trial, we had a group of just 10 people who got a second dose 4 weeks after their first dose, and they had a stronger antibody response. So then we changed the clinical trial and said everybody's getting 2 doses because we'll get a better response, so we'll get better vaccine efficacy. We were actually looking at efficacy after 2 doses, not after 1 dose then in the trial. But it was with a 4-week interval. We had delays in getting vaccine manufactured in time for these very large clinical trials that we're trying to do. And so some people did have a much longer interval between their first dose and the second dose in the trial. That was the group that actually had high efficacy in the clinical trial. And so we didn't have anything before 2020, and this information was gathered during the process of running those clinical trials that showed that that interval was best. In fact, if you wait even longer, it's even better, but you have to compromise between getting the strongest immune response and not waiting too long for the second vaccine. The half dose was a bit of a red herring. It was actually, you're right in that we saw very high efficacy in the people who had a half dose and then a full dose, but they also had a very long interval between the 2 doses, and it was really the interval that mattered, not the fact that they had a half dose first. And we don't want to give people a half dose for their first vaccine because their immune response is lower if they have a half dose. And that means in the period they're waiting for their second dose, they'll be less protected. So the best answer was to give 2 full doses, 2 weeks apart, very strong response to the first dose, and it doesn't lose efficacy for at least 3 months. And then you give the second dose, and it's even better and long lasting, and also broader, better at responding to the variants. once you get your second dose, it's even better after the third dose.
The girl in the, yeah.
Which special quality of yours helps you throughout the vaccine journey? What is your motivation or inspiration to become a scientist? What quality or set of skills a fresher need to become a scientist?
So what motivated me to become a scientist was that I always wanted to understand how things work, and my particular interest is biological systems. What makes biological systems, viruses, bacteria, mammals, what's going on at molecular level that's behind all of this? And so I tended to concentrate on the microorganisms because they're simpler and you can actually really understand completely what makes them work, whereas mammals are much more complicated. So I always wanted to study science, to understand how things work, but then how can we use that knowledge? How can we develop something that improves human health? So vaccine development works for me because you can understand a lot about how the immune system works, how the vaccines, the viruses that we use to make the vaccines, what to do with those, and then we can use it to make a tool that can improve human health. So that was what really motivated me to get started.
And what are the skills?
The skills, I think you have to be quite obsessive about the details. So when we're testing things scientifically, we have an idea and we think that this might work, but then we have to think about how we're going to test it and how are we going to know if this really does work and that it's not just some spurious result. And you have to obsess about what could go wrong. If you get an unexpected result in your lab research, it's often really interesting. It's often when something's got a bit wrong and, well, that didn't work out how I expected, that you can find something new. But sometimes there's a very silly reason why it's got a bit wrong. Somebody just mixed up the labels on the tubes and that's why you got an unexpected result. So you have to get into, why is this not giving me the result I expected? Let's repeat it. Let's think about what's behind all of this. And then sometimes, not often, but sometimes that leads to a new discovery. So for me, thinking about the details of exactly what's happening is really important, because if you just gloss over things, accept that, oh, it must have been an off day, we'll ignore that result because maybe the regime wasn't working that day. Maybe then you've missed something that's really important. So you have to continue to keep thinking about, why did this turn out exactly the way that it did? So it's quite a lot of effort. You have to be quite obsessive about the details.
Somebody at the back, right at the back, yeah, with the red, yeah.
Thank you, Sarah. Is the jury out about which kind of vaccine is that, the Pfizer or the AstraZeneca? The second point, which is more interesting to me, is that when the virus hit, different countries were affected at different rates, and we all thought Bangladesh was just going to be decimated, slums were going to be decimated, and nothing happened. I mean, compared to what was expected. And finally, I was in Australia when Australia banked on AstraZeneca, and then the blood clotting issue came out. And then we got delayed for another 12 months, so I don't know what I have to say to that.
Okay, so purely based on the efficacy data from the original clinical trials, the Pfizer vaccine has higher efficacy than the AstraZeneca vaccine. That was in specific countries against the early variants, and the picture becomes much more complicated. So it's really difficult to see, which is the more effective vaccine over long periods of time now that we have all the variants to deal with. We always said to people, it's not about competition between the different vaccine developers. It's about getting as many vaccines made as possible, as many doses of these vaccines. Because the whole world needs to be immunized. We need a lot of this. So it's not about who wins. Sometimes in drug development, it's who's first to market, they win everything. That was not the case here. We needed lots of vaccines to be made. And if everybody had been trying to make vaccines using the same technology, that would not have been good, because we had supply chain shortage, we have shortage of raw materials, we had difficulty getting hold of everything that we needed to make the vaccines, and if everybody was trying to use the same raw materials, we would have had even more supply shortages. So I think it's really about determining which vaccines satisfy the criteria for use, and then accepting that those are the vaccines that should be used. On the blood clotting issues, it's something that we haven't known about before we started the clinical trials. It's so rare that it was only when tens of millions of people had been immunized that it started to be, we started to get the data that this was happening. It only happens after people's first dose. If people haven't had that reaction after the first dose, it doesn't happen after the second dose. We also started to see that there's a geographical distribution, so the rate's highest in Northern Europe, lower in Southern Europe, and we don't see it in the global South. We don't know exactly the mechanism behind this, but it's a combination of both genetics and environment, and in some countries it wasn't seen at all. So Australia actually had a really good response to this in that they set up an app to track people after they've been vaccinated. So they wanted people to report back if they had headaches and things like this in the first few days after the vaccination. So the hematologist quickly discovered that if people did start to have symptoms, they could be treated, but you needed to know that they were having the symptoms. So that was the response that Australia had to it. But it's very rare, and it's the risk that nothing is ever risk-free, and not being vaccinated is far more risky than receiving the vaccines.
And the other question was the global South or certain countries, the prevalence was slightly less than one thought. Is there any thoughts you have on that? On the actually getting COVID, even without having a vaccination?
Well, I'm not sure whether the prevalence was lower or whether the severe disease and the deaths was lower. So I think, in Bangladesh is quite a young population, and where we're seeing the highest rates of mortality is in the old people with lots of comorbidities. And so if you have a population which is much more biased towards the young population, then you're not going to see so many deaths and so many severe cases. And we know that most infections with SARS-CoV-2 are mild infections. They can be asymptomatic, but what cripples healthcare systems is when you have enough people who are in hospital and requiring treatment that you have to devote all resources to that. And so even if the mortality rate is very low, it can still have a massive effect on the healthcare system. In a country where people are maybe a bit healthier in general, then you don't see that happening.
Right at the back. Yeah, that girl there, right? Yeah. Yeah.
Hello, I'm a student of microbiology, so I'm very starstruck to be here and listening to you. My question was, when new variants arrive, what changes in the characteristics of the vaccine do we have to update to deal with new variants? And another question is that we know that the mRNA vaccines such as Pfizer and Moderna needs to be stored in -7 to 80 degrees Celsius, but why do we, the Oxford vaccine doesn't have to be stored in such low temperature, and so we got it very quickly here. What would you like to explain about that as well? Thank you.
So I'm not quite sure what you were saying about the second question. The storage temperatures are different. Was your question about will there be more development for the RNA vaccines?
No, we know that the mRNA vaccines needs to be stored in lower temperature than the Oxford vaccine. So why doesn't the Oxford vaccine require that storage temperature? What characteristics of the vaccine allows that?
It's just a very different type of vaccine. It's based on DNA rather than based on RNA. DNA as a biological molecule is inherently more stable. DNA is what lasts throughout our lives. RNA is something that's made in our cells short term, quick turnover, so that we can switch on making a protein, and then we need to lose that RNA, so we switch off making the protein. It wouldn't work if RNA was very stable. So when you're using RNA as a tool to make a vaccine, it's still not very stable, and so you have to store it at low temperatures. They have enhanced the stability to a certain extent by making some chemical modifications, but there's just a major difference in the two types of vaccine, which means that it needs to be stored at low temperatures. There is some work on freeze drying it, which may help to keep it at higher temperatures. On the variants, Actually, the original vaccines are still working really well against the variants. And as I said, each time you get an extra dose of the vaccine, you get a broader immune response. We've seen that it's improved at the third dose, it's improved even more at the fourth dose. It's not really very helpful to try to switch the vaccine to match the new variant, because by the time you've done that and got that vaccine into manufacturing and rolled out, there'll be a different variant. And so we're never going to be able to have enough vaccines to use around the world against exactly the variant that's circulating now, because it keeps changing. If you think about the flu vaccines, surveillance all the time, and what goes into the flu vaccine each year is changed, but it's based on what was circulating last year. So we're always behind the curve. And actually, for the coronavirus vaccines, because of the type of vaccines there are, there's a good immune memory, good T-cell response. We get quite broad protection, and the original vaccines are still doing a really good job.
Yeah, here, the boy with the